I'm Sornambiga, Assistant Professor in Microbiology at SNS College of Nursing. Welcome to this session as acid-fast bacteria and 5-pillar concept followed by the SNS institution. In this presentation, we will explore how acid-fast training helps to diagnose diseases like tuberculosis and leprosy, and how our SNS framework enhances learning research and clinical excellence. Moving on this acid-fast bacteria with 5 pillars. That exploring the critical role of the acid-fast techniques to modern healthcare diagnosis, their pivotal applications of the SNS institution and that mycobacterial infection worldwide. Let's begin with the first pillar. The first pillar is understanding the acid-fast fastness. That acid-fast bacteria have that very special cell wall that thick, waxy and rich mycolic acid. Again I repeat, it is a special cell wall that thick, waxy and rich mycolic acid. Because of this is loss of colors and exposed that acid alcohol during the straining. This property helps us to identify the bacteria like Mycobacterium tuberculosis and Mycobacterium leprosy and Nocardia species. So this resistance to decolorization form of that scientific foundations that acid-fast training used with the microscopic labs. Here I am giving some examples for your reference purposes. Now let's move on to the second pillar, diagnostic staining techniques. We mainly used the three important methods. One is Ziehl-Neelsen method. This is a classic hot staining method. To heat helps to red dye in that Carbol Fuchsin and enter the waxy cell wall. After the staining, acid-fast bacteria appear bright red under the microscope. In this Kohn method, it is a cold staining version. It is use a high phenol concentration, so heating is not needed in this method. It is a safer and widely preferred in this labs. And we have to orange and Romaine method. It is a fluorescent under that UV. It is a method to sensitize the fast screening lodge in the samples. In the third pillar, highlight why acid-fast staining is important clinically. The tuberculosis still kill 1.5 million people each year globally. Quick and accurate detection saves lives. At SNS institutions, we have the frontline diagnosis network like nursing, allied and paramedicals. In the tuberculosis leprosy surveillance, by using the rapid microscopy method, we ensure early detection and timely treatment to prevent the diagnosis of the community in this healthcare. The next one is the fourth pillar, focusing on the biosafety. In the specimen handling, the backbone reliable result. We collect the early morning sputum samples or use the induced sputum to the patient who cannot cough in it up. And in special cases, we may use bronchoscopy or gastric aspiration, especially for the children. The very step followed in that biosafety protocol. Well ventilated room and N95 mask and gloves is proper specimen labeling. In that ensures the safety for the healthcare workers to accurate in the lab protocols. In that fifth pillar, it's all about integrated with that training. It is a SNS, continuously enhance the diagnosis method. We follow the protocol in the side by side and integrate with the molecular tool and expert in that TB fosters. To that, 5 pillar integrated with that standardized training programs and quality control programs, and in this cases technological integrated programs. That acid-fast training is not just a tuberculosis. It is also helps to detect the dangerous bacteria like Mycobacterium avium complex. And then the common in that patient HIV. The HIV patient also affected from their disease of TB. It is the opportunistic infection to the HIV patient. And the Nocardia species also causes lung and brain infection. Often it's mistake for the tuberculosis. Rapidly growing in the mycobacteria like that, Fatatium and Mycobacterium accesses found in that wound infection. To recognize these helps to the clinical giving the right treatment quickly by the way of Mycobacterium avium, Nocardia species and Mycobacterium. Some more species also there. I am here attached in that videos for your reference purposes. Next, here you can see the power of acid-fast staining. Before staining, a sputum sample looks like it is a debris. Nothing we can able to visible. But after the staining, it is a red shaped bacteria stand beautifully against the bubble. Against in that, it is the moment with the diagnosis we can able to see the bacteria by the way of very crucial thing in that microscopic identification and quantification methods. Finally, we have to play the vital role with the global TB control from diagnosis to treatment. Follow this through the five pillar concept: perception, production, and process. We ensure that highly quality diagnosis, biosafety, and continuous learning process. Together, we can stand to the guardian of world and combining traditional microscopic with the modern innovation to lies protect the community. And we have to conclude this. As I said, three P's like a precision, production, and process. It is one type of framework we have to follow in that measures. Thank you for watching. If you found this session useful, please like the video and insight from that SNS nursing. Thank you.